We're unable to hear the presentation right now, just so that we're all aware.
How about I unmute it? Thank you very much.
That's perfect.
So for those online, I'm just introducing ALS as a disorder. Unmute. Yeah, it's unmute. Oh, they
I
should.
can, yeah, I can hear you now perfectly.
Okay. We're on the same page now. Thank you very much. Great. Okay, so this is a highly heterogeneous disease, both at the age of onset, so it ranges from 30 to 70 years. Also on the site of onset, some people it starts in the legs, some people it starts in the bulbar region, some people it starts in the arms, and this has to do with what group of motor neurons start to degenerate first, and where the local muscle groups are affected, and also, highly heterogeneous rate of disease progression. So some people, the disease progresses incredibly rapidly, and other individuals, it can go quite slowly. And we'd like to understand what those factors are that are changing the rate of progression. I'll talk about the genetics of the disorder a little bit later in the presentation. About 10% of the cases are inherited. And from a therapeutic perspective, there's only one disease-altering therapy. This is an antisense oligonucleotide that targets one particular genetic form of the disorder with the SOD1 mutation. All the other FDA-approved therapies only slow the disease by a few months. So there's an imperative need to find new mechanisms of the disease and to try to leverage that for new therapies. Okay, if you look at the spinal cord of a patient that died of ALS, you can see on the right that there is rampant degeneration. This is actually staining for MHC2. We'll go in more detail. There's microgliosis. So if you look at the microglia on a healthy, quote unquote, healthy individual that did not die of ALS, they're ramified, they're extended, and on the right, you see these microglia, their morphology has changed, and they're chock full of dead and dying neuronal material. Individuals have tried to understand whether neural inflammation is a component of the disease, and they've used this PET imaging, Pittsburgh compound. This particular group, Nosmatase from MGH, found activation of neural inflammation as read out by this PET imaging in the primary cortex of these ALS individuals. And when they measured the level of neural inflammation by this marker relative to an individual's motor function, this ALS functional rating scale revised, they found that the individuals that had the highest levels of neural inflammation had the lowest motor function, suggesting that inflammation may be a contributor to the disease. If you look in the brains of individuals with ALS, you can also see evidence for extravasation of blood components. So this is hemoglobin, should normally be in your peripheral blood, and in this patient is leaking out of the vasculature into the brain. What are the causes of ALS? Well, clearly there's a contribution of aging. This is an adult onset disorder. Usually individuals get this in their 40s to 70s. So aging is important. There's some understanding of the environmental contributions, but still overall unclear in any individual, given individual. So we know that people who went to the first Gulf War were at increased risk of developing ALS, and this may have been associated with particular toxins that they were exposed to during their military service. There's evidence that cyanobacterial toxins, these non-canonical amino acids such as BMAA that are produced by cyanobacteria, can contribute to ALS in certain cases. There's an interesting epidemiological study in individuals from Guam that this was thought to be the case. There's also increasingly strong evidence that lead and polychlorobiphenols that accumulate in the brains can be a cause of ALS, but really the environmental contributions are largely uncertain. As opposed to that, the genetics are substantial, quantifiable, and approachable from a research standpoint. So if you look at ALS as a whole, about 90% of the cases don't have a clear familial history. But in about 10% of the cases, we can track within these families where a particular mutation in genes is sufficient to drive the disorder in the individuals. And you can see just by this plot that here's some of the common genes that are associated with risk for ALS. And these genes almost exclusively act in an autosomal dominant fashion, where you have a mutation on one allele, and that allele by itself is sufficient to cause the disease. And I'll be talking about this mutation in chromosome 9 open reading frame 72, the most common genetic contributor to ALS. in more detail. But before I go on to that, I just wanted to pause and have a sort of theoretical discussion of how these genes may be causing neurodegeneration and neural inflammation. I've put a couple hypotheses up on these slides. So we know that gene mutations by themselves are sufficient to cause neurodegeneration. And we know that when neurons start to die, they contribute to neural inflammation. And it's conceivable to think that this neural inflammation may be acting in a feedback mechanism to propagate neurodegeneration. But what's the contribution of systemic inflammation? Is it kind of an epiphenomenon that just kind of happens and has no contribution to the disease? Might it be acting in parallel with neural inflammation to contribute to neurodegenerative disease? Might there be a more linear relationship where systemic inflammation may precede neural inflammation that then contributes to neurodegeneration? So when we think about these, I'm sure everyone in the audience can come up with an opinion about which one of these hypotheses they think may be most similar to the what actually occurs in patients. And I would caution that it probably changes depending on what the causal gene mutation is and how it's acting. And really, I don't know what the answer is. The only way to really know what the answer to this question is to have disease-modifying therapies that target one of these branches to show that it actually is important in the disease. And here I would point to this drug, masitinib. So this drug targets a tyrosine kinase receptor that functions in microglia and mast cells. So it targets both the neural inflammation and systemic inflammation. And this drug was able to reduce risk of death over 2 years in in ALS patients by 47%. This led to about basically a 26-month extension of lifespan, which is a large change in the disease course. So there is evidence that targeting the immune system and inflammation can have a measurable effect on ALS. So for the rest of the talk, I'm going to focus on this particular genetic cause of ALS, this mutation in chromosome 9 open reading frame 72. So what is the mutation? It's a repeat expansion in a non-coding intron of the gene, a hexanucleotide repeat expansion, GGGGCC. So in most of us with a low risk, there's a few copies in this intron, but in these unfortunate families, this has been expanded hundreds to thousands of repeats. And what is the consequence of this mutation? So one consequence is that it reduces the transcription of the downstream coding gene. So we know more about what this gene does. It forms a complex with 2 other proteins that functions as a GTPase activating protein, has been implicated in endolysosomal vesicle trafficking, among other aspects. And so there's this loss of function hypothesis. But this repeat expansion is also transcribed. So we have long repetitive RNAs that get made, and then those long repetitive RNAs get translated in a non-canonical fashion into repetitive dipeptides in all reading frames. So you have these dipeptides, GR, GP, GA from the sense strand, or PR, GP, PA, that get made, long repetitive peptides that the cells now have to deal with. And they've been shown to be toxic in neurons when overexpressed at high levels. So this kind of gain of function hypothesis. And so a lot of work has gone to trying to target these toxic proteins made by this repeat expansion. And so several drug companies, among them Biogen, have developed antisense oligonucleotides that target so far the sense strand to knock down these dipeptide repeat proteins to see if that can slow the progression of the disease. But unfortunately, when in large clinical trials, these had been administered to individuals with the CNR72 ALS mutation. While it was well tolerated generally, it didn't have a beneficial effect in the patients. So even though it reduced GP, this marker of the sense strand, it had an elevation of neurofilament and cerebrospinal fluids, which is indicative of more neuronal death. And there was more functional decline in people who got the highest dose. So this has really caused some questions in the field about what the most important disease-causing mechanisms are from this repeat expansion. And it's really, I would say, renewed interest in what loss of function and how that might be contributing to the disease. So again, this protein downstream of the repeat expansion forms a complex with 2 other proteins, SMCR8 and WDR41. The gene is highly enriched in the motor neurons that degenerate in the disorder, but also in other myeloid lineages such as microglia, dendritic cells, and macrophages. And this complex has been associated with many functions: vesicle trafficking, autophagy, lysosome, mitochondria, actin cytoskeleton, and cytokine production. And our work in particular has implicated a role for this gene, at least in mice, for preventing the onset and progression of systemic and neural inflammation. So I'll talk about that in more detail. So when this gene was discovered as the most common cause of ALS, we generated mice that had knockout of this. So we replaced the mouse ortholog with this neomycin lax-E cassette, and then we made large groups of these mice. The first question we asked with this allele is, using the lax-E reporter, what cells are expressing this? And we looked in the spinal cord. This was work by Naoki Suzuki, and he showed that the large neurons in the ventral horn of the spinal cord expressed this gene. However, when he counted it in the heterozygous or the homozygous mutant mice, he didn't see changes in the numbers of these neurons, which suggests that loss of this gene is not sufficient in and of itself to cause motor neuron degeneration. But we did find is that these mice were at increased risk of death. So if you lost one allele in the blue line or both alleles in the red line, these mice died relative to their wild type. And this was due to their underlying autoinflammatory disorder, which I'll show you in more detail. We looked in the plasma using a multiplex Luminex array, and we saw that many different cytokines, pro-inflammatory and anti-inflammatory cytokines, were elevated in the mutant mice. And you can see that this is a log scale. And then we looked in the spinal cord using confocal imaging. And here I'm zooming in on a region of the mutant spinal cord. And you can see in green this CD45. It's a general marker of hematopoietic cells. And in red, IgG, so mouse immunoglobulin, that normally should be restricted to the vasculature, but here in this mouse you see is kind of leaking out. And what we found was that these peripheral immune cells were actually leaking into the spinal cord at these sites of blood-brain barrier disruption, and then also present within the spinal cord parenchyma. We quantified this with mass cytometry. It's a method very similar to flow cytometry with antibodies. And we found that while the total numbers of microglia didn't differ within the mice, we did see accumulation of neutrophils and CD4 T cells in these spinal cords. We were not the only people studying this allele. And there's actually some surprises when we showed our data to other groups. So whereas we saw that the heterozygous and the homozygous mice die at Harvard, and Johns Hopkins saw a very similar pattern. At UC San Diego, they only saw the knockout mice die, but the hets were relatively protected. And at Cedars-Sinai, they showed no death at all. And all of these groups were studying the same loss of functional allele on the similar genetic background, the same genetic background in many instances. So what is causing this? And we were particularly interested in this differential disease manifestation because in patients, there is incomplete penetrance. So here's an example of a human pedigree. So this is one of these families. And so you can see some individuals within this family developed frontotemporal dementia, a related neurodegenerative disease that we now know is on the same clinical spectrum as ALS. And some individuals developed ALS. So this mutation, it demonstrates, is sufficient to drive either FTD or ALS in the same families. And there are some individuals who develop symptoms of FTD and ALS. But there's also these obligate carriers, these parents that we know must have had the mutation but didn't die of ALS or FTD. And the paper notes that some of these individuals lived to their 90s. And so you say, why didn't those individuals develop the disease? Right. And so if we could identify these modifiers of disease that would protect some or cause the disease to become more rapid in other individuals, these could lead to new therapies. So we wanted to test the contribution of environment to disease in this mouse model. So we continued to breed our mice at Harvard, but then we aseptically re-derived the mice at the Broad Institute a few miles over in Cambridge, and then made large cohorts of these mice for survival in age-matched tissues. And this is published, so I don't want to go on this too long, but basically we recapitulated the survival differences that we found at Harvard, but when we put them at the Broad, they're basically completely protected. We measured different aspects of their inflammation. So this motor deficit was protected, premature death was protected, and less neural and systemic inflammation when we reared them at this other environment. We made a large cohort of mice. We tested them on the rotarod, this measure of motor performance. We bled them, and then we started gavaging them daily with antibiotics, and then measured the outcome in the disease. And we showed that the mice that received antibiotics to target their gut microflora improved in their motor function and reduced markers of inflammation in the blood, neural inflammation. When we looked in the spinal cord of these mice, again, confocal imaging, we see those CD45 high peripheral immune cells infiltrating the vehicle-treated mutant mice, significantly reduced in the antibiotics-treated mice. And we looked at microglia with this marker of microglial activation, dectin-1. You see an example of this mutant microglia reactive here and quantified here on the right, and we see reduction of microglial activation with this antibiotics treatment. So this is showing us that signals from the gut are contributing to neural inflammation in this model. And we've done many other studies with fecal transplants as well to show that it's not just a off-target effect of the antibiotics. Here's an example of the peripheral blood cytokines. So cytokines elevated in the mutants treated with vehicle and significantly reduced by antibiotics. We want to understand what contributors from the gut might be causing these. So we took fecal matter from our mice, and we also asked the ALS community to send us fecal matter from theirs, institutions where the mice got sick, institutions where they were protected, and we sequenced it. And we found that, so here in the middle, a principal component analysis with each one of these dots being a fecal sample from one of these mice in the different institutions, that whereas the fecal communities were most similar to one another within an institution, the largest contributor to variance, principal component 1, actually separated the environments where the mice got sick and the mice were protected. You can see it on the right in this Bray-Curtis dissimilarity complex. So the bacterial communities were different at these different institutions. We did a lot of work. This is just a summary of that work, that these mutant mice develop autoimmunity, inflammation, behavioral deficits, and death. They have peripheral immune cells infiltrating their central nervous system and microgliosis. But if we change the gut microbiota, either by changing the environment in which they're reared, treating them with broad spectrum antibiotics, or giving them a fecal transplant from one of these environments where they're protected, we can recover all of these systemic and neural inflammatory phenotypes. So the question that we've gone on since then is, well, what cell types is this gene functioning in, and through what molecular mechanisms? I told you that we're not the only people studying this. Many people have implicated roles for this complex in several facets of cell biology. So you can see in the top left that this is involved in many different cellular processes, autophagy, vesicle trafficking, stress granules. In motor neurons, it's involved in actin cytoskeleton at the synapse. In microglia, it's been implicated in clearing aggregated proteins and phagocytosis and cytokine production. That seem to be related to its ability to control these small GTPases. That's kind of what it was known. We had done bone marrow transplants where we take the marrow from wild type or mutant mice and put it into wild type or mutant animals. So you see that the wild type mice that received wild type marrow survived, and the knockout mice that received knockout marrow died. And if you gave a knockout mouse wild type marrow, you could extend its lifespan. And if you took a wild type mouse in teal and gave it mutant marrow, you could kill it. So we know that the gene is functioning in bone marrow derived cells, which suggests hematopoietic cells. We wanted to study this through a different mechanism. So we got a conditional mutagenesis allele from the Pasteur Camp group. So, the C972 locus, exons 4 and 5 are flanked by these LOXP sites, so that when you cross this with pre-recombinase expressed in particular cell types, you can cause removal of those 2 important exons, leading to premature stop codon. So we can conditionally mutagenize this gene in lineages, and we cross this conditional allele to 7 different pre-recombinase lines. 2 lines that recombine in hematopoietic stem cells to knock it out in all hematopoietic cells, and the ones that either knock it out in the myeloid lineage, the lymphoid lineage, or specific components of the lymphoid lineage. We looked at splenomegaly, this just general marker of systemic inflammation in these mice. We saw that if you knocked it out in all hematopoietic cells, VAV1 or MX1, you got splenomegaly. And in the myeloid lineage, you are sufficient to drive that. So some aspects of this model are driven by the gene acting in myeloid cells, whereas we didn't see that with the lymphoid cells. And when we made large cohorts of these mice and tested their survival, we saw that indeed, if you knock the gene out in the myeloid lineage, it was sufficient to kill the mice. But when we looked at other facets of this model, in particular, levels of plasma IL-17 in the plasma or neutrophils circulating, we showed that if you knocked it out in all hematopoietic lineages, you got these phenotypes. But if you only knocked it out in myeloid or lymphoid, you didn't. So it seems that there are, the gene is functioning in multiple lineages in a synergistic fashion to drive these really bad immunological outcomes. We want to understand, well, what was the gene doing? So here we employed mass cytometry again on the spleen. And we identified all of the different hematopoietic populations that you would expect in the spleen. So macrophages, dendritic cells, monocytes, neutrophils, T cells, and B cells, using these characteristic cell type surface markers. But then the power of this technology is that we could then look at the expression of other molecules on the surface of these different lineages. I don't expect you to know what all these are. I'll actually introduce them on the next slide. But what really jumped out from our analysis was that the macrophages from these mutant mice at a young and an old age had consistently high levels of this molecule CD80. So what is CD80? So this is a co-stimulatory molecule. So, if you bring out your immunology textbooks, we know that antigen-presenting cells, such as macrophages and dendritic cells, survey their environments for pathogens and other inflammatory mediators, and they present degraded molecules on their major histocompatibility complex to T cells. But if you just get a T cell activation through MHC, they'll become anergic, they'll turn off. You need a second signal provided by these co-stimulatory receptors, CD80 or CD86, that bind CD28 on the naive T cells to say, yes, you should proliferate in response to this antigen. And then you get cytokines to say, what type of T cell should you become? So these macrophages seem to be giving too much co-stimulation. We wanted to know, we found that in the spleen. Is it happening in the central nervous system? So we looked at the spinal cord and brain of these animals, and indeed, we saw that forebrain microglia had elevated CD80, and in the cord and in the forebrain monocytes had elevated CD80 in the mice, in the mutant mice. So here, we said, okay, great. We found something about mice, but are the mice really relevant to the ALS patients? So here we collaborated with Rodrigo Lopez Gonzalez at Cleveland Clinic Learner Research Institute, and we got spinal cord sections from 4 human ALS patients and 3 non-ALS controls, and we stained them for CD80 and IBA1. So here's our confocal imaging. So you see IBA1, microglia, and CD80 co-localizing in these CNR72 ALS patient microglia, and not as much in the non-ALS. Video tells 1,000 words. So here we have this microglia in green. See the CD80 staining in the human ALS. We'll zoom in on that microglia. See the CD80 staining in magenta. And if you take the microglia away, you can see CD80 kind of mapping to those processes. And then if you look at the control on the right, vastly less CD80 in these spinal cords. So we quantified this. We looked at more than 440 microglia per case, and we found that there was significantly more CD80 in the microglia from these ALS patients. Next, we wanted to understand a little bit more about the mechanism of how this gene might be controlling CD80 in these cells. And so we thought, we knew that bacteria were important for this model, and so we wondered, well, what signals from bacteria might be driving this? So again, our immunology textbooks, we know that myeloid cells recognize components of the environment through these toll-like receptors. They're expressed either on the cell surface or within the endosomes. And when these get activated in response to conserved molecular moieties present in virus and bacteria, they activate pro-inflammatory signaling cascades, MAP kinases and ERK, to drive inflammation. So we edited, using CRISPR-Cas9 ROT264.7 mouse myeloid cell lines for CNR72. And we applied these synthesized TLR agonists, toll-like receptor 2, 7, 4, or 3. And then we did flow cytometry to look at CD80 on the cell surface or MHC2. And I know this is a lot, I'll try to walk you through it. So if we gave the mutant cells PAM3 to activate TLR2, we saw the mutants had much more CD80 on their cell surface. Also, if we activated TLR7/8 using R848, we saw more CD8 on the cell surface. Whereas if we gave LPS, we did see an elevation, but the mutants didn't perform better than the wild types. And we really never saw differences of elevation of MHC2 on these molecules. So there's some way in which the cells are recognizing particular toll-like receptor agonists to lead to CD80 accumulation on their cell surface. We use Western blot to look at this in more detail. So if we just blot for CNR72, you see that we did a good job of killing the gene. And if we don't stimulate the cells with vehicle, we don't really see much CD80 in the cell lysates. But in the mutants, when we give that TLR2 agonist, we see accumulation of CD80. If you look at immunofluorescent images of these cells, again, unstimulated, really not that much staining. A little bit more staining in the vehicle PAM3 stimulated wild type cells. But in the mutant cells, you see much more accumulation. You actually see it around the outsides of the cells. So this gene seems to be suppressing the trafficking of this molecule to the cell surface. This is a model that I proposed based on the work that we have. So we believe that perhaps the CNR72 complex is promoting this molecule to go to the lysosome under normal conditions so it can be degraded and prevent it from the cell surface. But in the absence of this gene, you have less CD80 going to the lysosome and more CD80 going to the cell surface. But what about other stimuli? Is this just in response to bacteria? How about cytokines like those that are circulating in these animals? So we took recombinant proteins, recombinant interferon gamma and recombinant IL-17, these pro-inflammatory cytokines, and we applied it to our macrophages. And again, if you don't stimulate them, they have very little CD80 on their cell surface. If you give interferon gamma alone, it's sufficient to drive accumulation of CD80 in the mutants. And this is exacerbated by IL-17 with that purple arrow you can see there. So it seems like IL-17 is working in conjunction with interferon gamma to drive this molecule to the cell surface on myeloid cells. But here's another model. We think that microbial stimuli being recognized by macrophages and microglia, it's driving the production of cytokines and CD80, and CNR72 normally prevents the accumulation of CD80 on the cell surface, but in its absence, there's this chronic co-stimulation. It may affect the decision of T cells to adopt these pathogenic Th17 identity. And then when those cells start to produce interferon gamma and IL-17, it acts in a feed-forward mechanism to really exacerbate this phenotype. I just have one more experimental series to show you. We really wanted to test whether this was important in the mice. So we made a new cohort of mice, and then we treated them twice a week with an antibody that blocks IL-17. And we measured them on the rotorod and took blood measures, and then took these mice down. This data is just to show you that these mice were old, about 7 months old when we started the trial. They performed poorly on the rotorod to begin, and they had systemic inflammation as measured by neuroinflammation, balanced cohorts. And then when we started treating these mice, we saw both females and males that we were able to improve the motor function to a greater extent in the mice that received IL-17 neutralization in orange relative to the mice that received the IL-17 neutralizing antibody in red. We improved motor function. When we looked at neutrophils in the peripheral blood, the mutant females that got the isotype in red continued to get worse, but the ones that received the blocking antibody did better significantly, although we didn't make a difference in males, and we don't completely understand this. And then we looked in the spinal cord of these mice, and we measured markers of inflammation that we had identified by sequencing. We saw that the vehicle-treated mutants had higher levels of these inflammatory genes, and that these levels weren't different from the wild-type mice when these mice had been treated by anti-IL-17. So really to wrap this up, we've shown that this gene is acting in both myeloid and lymphoid lineages to prevent the accumulation of IL-17 in neutrophilia. This gene seems to be opposing trafficking of CD80 and its co-stimulatory activity in response to bacterial moieties and these cytokines produced by pathogenic Th17 cells. And we've shown that IL-17 neutralization can improve motor function and limit neural inflammation in this model. We've shown the CD8 is enriched in human ALS spinal cord. So what's the evidence that this molecule may actually be important in ALS patients? So several groups have looked at levels of IL-17 itself in patient fluids. So if you looked at the serum, one group showed that it was elevated in sporadic ALS patient serum. Another group looked at even more individuals, 50 to 100 individuals, and they found that it was elevated both in the serum and the cerebrospinal fluid. And if another group looked particularly in these CNR72 mutant patients, and they saw that it was most highly enriched in those cerebrospinal fluid. So the molecule is there. If you look at circulating Th17 cells, these cells that we know are primary producers of IL-17, groups have shown that they're enriched in the peripheral blood and that they correlate inversely with motor function. So if you have the highest levels of Th17, you have the lowest motor function. Look at neutrophils. We know neutrophils are driven by IL-17. Neutrophils are up in ALS patients in the blood, in the cerebrospinal fluid, and the patients that have the most neutrophils died the fastest. And from a patient perspective, we don't even need to develop new therapies. there are already multiple drugs that target this pathway that have been shown to be beneficial in rheumatic diseases like psoriasis and rheumatoid arthritis. So I think that the ALS community should be considering whether we should be using these drugs to treat these patients. With that, I'll wrap up. So we know that individuals have this variable genetic risk at birth. In some instances, it might be a very high risk due to causal mutations such as the C9orf72 mutation. We think that inflammatory microbes in our gut may be contributing to this risk of developing neural inflammation and autoimmune diseases. This interaction between antigen-presenting cells and T cells and co-stimulation may be driving these neural inflammatory and systemic inflammatory effects. And IL-17, may be promoting this early and perhaps sustained effects on neural inflammation in these individuals that could contribute to disease onset and disease progression. So with that, stop. I want to thank many members of my group at Case Western Reserve. A lot of this work was done at Harvard while I was a postdoc with Kevin Egan, in particular Francesco Limone, who's now a postdoctoral fellow at NYU, and Alex Couteau, who's now a graduate student at UCF, and our collaborators at UCB, Case Western, LRI. And with that, I'll stop and be happy to take any questions.
I have a question, but I'll wait until everybody there asks theirs.
Is that possible? You from the Zoom, would you like to ask your question?
Sure. Thank you so much for this presentation. I'm CJ, one of the residents, PGY5 for Child Neuro. So my colleagues, Jamie and I and Dr. Barnes are working on sort of the neuroinflammatory axis as it relates to autism. And we find a lot of parallels in the biomarkers and cell types that you're discussing here with ALS. So I was wondering, what we notice in autism is that like the Th17 cells are going to be upregulated. IL-17A is upregulated. And then Tregs, generally is going to be downregulated. Like there's less regulation of inflammation and more sort of feed-forward inflammation occurring in children with autism, especially in certain subgroups. So sounds like, and that dysbiosis also occurs in a certain subgroup of children with autism. And these microglia, basically the gut microbiome is dysfunctional, and it results in a feed-forward inflammation that signals to microglia to start pruning and screwing things up. It sounds kind of like you found a similar situation here with ALS. I was just wondering if there's any evidence for literal clinical dysbiosis in patients with ALS, like if they have constipation, diarrhea, or sort of changes that occur, or some sort of signaling, or even fecal sample differences to the diversity of microbacteria in the gut that may, be a good way to screen patients and say, hey, you are at risk. You have a higher, not just higher IL-17, but also evidence that this is due to the gut, that we can maybe find better subgroups and to provide these immunotherapies. So as a long-winded question.
Thank you. Yes. Okay. So, I would defer to my clinical colleagues to talk more about their personal experiences treating ALS patients but from what I've read, there are changes in the motility of the gut, the motor neurons that innervate the gut, that can lead to changes in the rate at which the gut is cleared in those individuals. One of your questions was, is there evidence for dysbiosis in ALS patients? So several groups have tried to look at this so far, and the results are mixed. So one group from Germany looked at 25 sporadic ALS patients and 25 healthy controls. sporadic ALS patients that had no history of other autoimmune or inflammatory diseases. And when they did 16S ribosomal DNA sequencing, they didn't find differences within those gut communities. But other groups, a group Aaron Elanov from Israel had sequenced different ALS patients, and they found changes in the community structure of their cohort of ALS patients. And another group, James Berry and Catherine Nicholson at Mass General Hospital, looked at fecal samples from ALS patients, and they saw differences in the community structure of ALS patients versus controls and matched non-ALS neurodegenerative disease controls. And they identified particular bacterial species, ones that produce short-chain fatty acids, and also Prevotella, that were significantly different between the ALS patients and controls. So the short answer is that there's not a good answer. We don't know specifically whether this is a consistent signature of the disease. And what's really important is, are these just correlative analyses as they stand right now, or is there a mechanistic understanding? Is there causality? How are these microbes interacting with our cells in our genome to drive neural inflammation and to drive that risk? And that really we don't know yet. And what we need to do is we need to take fecal samples, microbes from patients, and put them into animal models that are susceptible to influences of the gut microbiome, and use that to try to identify which are the actual microbial communities that are sufficient to drive neural inflammation, and how are they doing that, right? And so really that is another aspect of the lab that we're driving forward right now.
That's fascinating. If you want some more reading for later, if you've already checked out the RAGE system, or advanced glycosylation end products, it's worth just reading a little bit of review of RAGE and AGER. You may find with ALS to actually come up with some very relevant information, hopefully, for your research.
Great, thank you.
So if there are no other in-person questions, I have a question for you. It was a fantastic talk.
Sorry for the online. There is a question being asked right now. We just don't have a speaker that can project it. So I'll restate the question to the online audience.
Fantastic, thanks.
Yeah. So if I can restate the question from the audience. So we're trying to understand, knowing that ALS is defined by degeneration of both upper motor neurons and lower motor neurons, and this is really a disease of circuitry. Do we understand completely why certain neuronal populations are more susceptible? And is there a difference in the temporal aspect that might help us understand why in some individuals you get lower extremity onset first, or upper features of the disease? Okay, so a lot of groups are trying to understand, what distinguishes motor neurons from other neurons in our body and why they might be more susceptible, and whether there is a difference between the upper motor neurons and the spinal cord motor neurons. So one technical limitation, people are doing single cell sequencing on brains from ALS patients and frontotemporal dementia patients relative to controls, and they are finding that the upper motor neurons have higher levels of the genes that, when they become mutated, are sufficient to drive the disease. So that would support a hypothesis in which the motor neurons, at least in the brain, are more susceptible to these toxic effects of these gene mutations, because they perhaps are more dependent on those genes themselves. But we haven't done a good job of sequencing the spinal cord motor neurons because there's more myelin in the spinal cord, and it's just so far has been very technically challenging to do sequencing on the spinal cord motor neurons. So there hasn't been a good comparison from gene expression perspective of what distinguishes upper motor neurons from lower motor neurons. There's some evidence in mouse models and using human-induced pluripotent stem cell derived motor neurons that spinal cord motor neurons and upper motor neurons express particular genes like matrix metalloproteinase 9 that may confer risk to them relative to other motor neurons that are spared, like those that innervate the eyes and the lower gastrointestinal tract. But it's really unclear. And to kind of take a step back, what's the role of inflammation in this? Is the brain going to be more targeted in some individual relative to the spinal cord? And would that differential activation of Th17 cells or microglia in that particular person drive why the upper motor neurons might degenerate first, or vice versa, the lower motor neurons? And that's really something that we don't completely understand. Maybe I can bounce to the Zoom question and then come back to one more from the audience. The person who on Zoom had a question.
Yeah, this has been a great talk. Taking a step back from the details, the idea of neural inflammation and the things that we believe can influence neuroinflammation, what about things like exercise and activity or inactivity? Things like diet, so high-fat diet versus a ketogenic diet, chronic pain syndromes, things that we know can influence CNS inflammation. Does that influence these pathways and the presentation in your models? Did you hear the question? Our sound seems to be off.
Yeah, I'm not sure why we can't hear you anymore.
I could hear you, David.
Some people think that overactivity may be a contributor to ALS, that the motor neurons themselves are more stressed and they have higher energetic demands, that this may be a risk. But then, yeah, diet and lifestyle, how that might contribute. Is there ALS patients, once they start to, have the features of the disease, it's more difficult for them to swallow. And this can lead to changing patterns in their diet, what they're capable of eating. And so once the disease has onset, how might dietary changes and, changes in the gut microbiota then perhaps contribute to the rate of disease progression in those individuals? I don't think we have a great understanding of that yet. A question from the audience? Yeah. So if I, yeah. Okay. Let me see if I can phrase the question for the audience in the Zoom. So we know that the C9orf72 mutation is sufficient to drive ALS and FTD, but IL-17 as a pro-inflammatory cytokine contributes to many systemic and neural inflammatory disorders. And where is the specificity? Why would IL-17 be important for these particular diseases? And should we be focusing primarily on the motor neurons, which are really the big cheese in the disease? Okay, let me talk to this from a couple perspectives. So the C9orf72 mutation can cause many neurodegenerative conditions, so not just ALS and FTD. Individuals get Parkinsonism, they get Huntington's disease like phenocopy, and in some instances, memory loss like Alzheimer's disease. So the clinical manifestations of this mutation are even larger than ALS and FTD. It's just somewhat more rare. Yes, IL-17 can contribute to systemic inflammatory diseases, and I really don't have a good answer for why it might particularly put people at risk for ALS. I guess in my own mind, I'm thinking that it might just be a governor, and it might be increasing the intensity of neural inflammation within these individuals. So an underlying motor neuron specific degenerative process might then be exacerbated by IL-17. And from a therapeutic perspective, you don't necessarily need specificity. If that molecule is contributing to the disease progression in those individuals, you can target it and block it. And maybe an IL-17 neutralizing therapy might be beneficial for multiple neurodegenerative diseases. And then the last part of your question was, well, why C9orf72 and why specifically ALS? So the gene certainly is expressed in motor neurons. It doesn't seem to be required for them to be alive, but many groups have studied this and show that synaptic biology, breakdown of aggregated proteins is not as good in neurons that have loss of function. And when you take cells or you take mice and you put these gain of function and loss of function effects together, you get even worse phenotypes. So this gene and that complex seems to be mitigating the most toxic effects of the gain of function aspects of the mutation. And how those motor neuron specific effects are interacting with microglia and the role of the gene in microglia. And our data seem to suggest that the C9orf72 gene is controlling this co-stimulatory molecule and how that's shaping response to degenerating motor neurons. Might be an important aspect to go further.
Is there a question being asked?
There's a question being asked from the audience right now.
Sorry.
Okay, the question is, should we be treating the disease with radiation to broadly target the hematopoietic system? Yeah, this has been tried in some sporadic ALS patients. It didn't work. And patients have also been treated with broad spectrum immunosuppressants, which didn't work. Those therapies weren't specifically designated to individuals with a C9orf72 mutation. So you could argue that perhaps this might be more relevant in individuals with that mutation. But I think in the end, the answer is going to be that the immune system is important as well as detrimental. And so it's important for clearing toxic aggregates and supporting neuronal health. And if you just get rid of the immune system or suppress its activity, it may exacerbate some aspects of the disease and prevent it from being beneficial. Yeah, so bone marrow transplants as well. Stanley Appel did bone marrow transplants in sporadic ALS patients, about 7 of them, maybe 10 years ago, and it didn't have a measurable effect. And bone marrow transplants is really toxic, hard thing for patients to deal with, especially when they're dealing with the symptoms. One more question from the audience here. Yeah, many people. So the question was, I've kind of proposed this inflammatory mediated death of motor neurons, but can we rule out a kind of apoptotic or other form of cell death of the motor neurons themselves? And so to that, I would say that many people have studied how motor neurons die, particularly with the SOD1 transgenic mice that have been studied in many, many years. And those studies seem to support a cell intrinsic apoptotic RIPK1 dependent cell death. But if you get rid of all of those receptors, the caspase-8, the RIPK1, all of the ways in which the cell is capable of inducing a cell death, but you continue to force the cells to make toxic SOD1, they turn into zombies. The cells are still there as shells, but they are incapable of functioning, and the animals still die. So, it seems like they're in different mutations or sporadic individuals, the motor neurons may die from different causes, but death or just being non-functional is going to have the same cause in a patient's and paralysis. So really trying to understand what those upstream activators are that are causing the cells to die in the first place. Are there any other questions from the Zoom audience before we wrap up? Thank you very much, everyone.